Welcome to another Wednesday of AP Bio. I'm so glad you're all here. Oh my goodness, we've got 12 tonight. Oh, I think this is my new record. I'm so excited. Yay! Welcome, Zara, Madeline, Sandra, Jess, Ariana, Panda, Delwyn, Kelvin, Neri, Wendy, Sam, Sarabia, and Consuelo. I'm so glad you guys are here. Woo. For those of you who are new, because I see several of you are, so whoop. My name is Jessica Nadsam. I am an AP biology teacher in not Southern. I'm in the South. I am in Arkansas in the Delta region. So I've been doing this for about 5 years now. So I'm not new to this, but I consider every moment a learning moment. So let's put it that way. For example, I thought I was really good and all signed in, and then I realized, oopsie, I never actually turned on my slides, but they're on now, so we're good. So welcome to Fiveable. Hopefully you've heard the great news that we just sent an email about. We strongly believe that all students should have the opportunity to do well on their AP exams and to exceed, because that's a gateway to college. And as a result, all of Fiveable is now free, as in this replay that you're watching. If you're not watching it live on February 19th, 2020 at 7 p.m. Central, it's free to you now. You don't have to pay for it. So whoop, whoop. Hope you guys are excited about that. We will still have some premium options called cram passes. So if you're interested in like some extra help for a certain subject, you should have gotten an email about that. But just wanted to celebrate that because whoop, whoop, equitable access. I would renegade, but I can't renegade. I can only do like mom dances. So that's what I got, y'all. All right, but it's already been 2 minutes, and you're probably ready to see some biology because that's what you came here for. So why don't we go ahead and get started with that? So I'm going to open this little thingy-majig, and I'm going to go to this thingy-majig, and whoop, here we go. La-da-da-da-da. So, whoop, whoop. It's a me. So here we go. This thing, we're going to talk about DNA replication, transcription, and translation tonight. Lots of topics, but a lot of stuff. I wish I could say we're just going to breeze right through it. And it's not that it's hard. So for those of you who may have already done this, if this is a challenging topic for you, I want to affirm you in that wrapping your head around it takes a minute. But once you get it, there's this light bulb moment, and you're like, this is why biology makes sense. And it's so cool. So that's what we're going to look at now. So that's what you're here for. Let's talk about it. I also want to highlight, one of the cool things about us going free with our content is that we will be able to put it everywhere. So you'll be able to find it all over the place. It's going to be awesome. We really hope you enjoy that. So definitely follow us. There's like little subscribe buttons and stuff. You go click, click, and then you're like, you are subscribed. And it's great because then you know when all the stuff comes through. And I don't know about y'all, but I like to be informed. I don't like when I don't know what's going on. And this sort of thing will definitely help you stay in the know about what you need to know for your AP stuff. All right, here's the things you actually came for. AP biology. So like I said earlier, we've got 3 topics. First one is DNA replication. With DNA replication tonight, I really do want to talk about the actual DNA structure and make sure we really got cell cycle locked down. If you don't have those, DNA replication can be really, really tricky. I'm going to be perfectly honest. For me, DNA replication is harder than transcription and translation. But that being said, it doesn't mean it will be for you. I just want to prepare you that there's a lot of terminology. And for a lot of students, they've never heard this terminology before. But AP requires you to have it, so we got to do it and da-da-da. So you're going to be experts by the end of the hour. That's what matters. So yes. Boop, boop. Then we're going to get into the central dogma, aka gene expression, aka protein synthesis. It has 3 different names for one cool thing. It has 2 steps, transcription and translation. We're going to get into those. You can see we've got a kind of like itty-bitty diagram down here where we take DNA, we turn it into RNA, and then it turns into mRNA, and then it becomes a protein. And we like proteins because we use proteins to get muscle and to make our skin and like cell signaling. And I can't even list all of the things because we would be here for the next 54 minutes just talking about that. And y'all want to do more than that. So we're going to move on instead. So let's get started with DNA. Want to do a quick recap on nucleic acid structure. If you don't have this structure already locked down in your brain, this is for you because I need to make sure you get it there and you keep it there. So nucleic acids, what are the two types of nucleic acids? You can throw it in the chat box. Two types of nucleic acids. What are they? I see 14 people. One of them has to have the answer in their brain. I'm so excited y'all are here. Woo. What are the two types of nucleic acids? Give me a hint. They're both 3 letters. Two types of nucleic acids. DNA and RNA. Awesome, Zara. Air high five. Great job. Whoop, whoop. So DNA and RNA are the nucleic acids that we're going to be talking about here. And you can see the nucleotide here. Now, this is a very, the best word I can think of is extra diagram. It's got a lot of extra vocab you don't need. But I did like it that it was so descriptive because I know some people really like to know the extra stuff. So I went ahead and threw it in there. So you're going to have your pentose. That means 5 carbon sugar no matter what. But one of the things AP likes you to remember, and I've seen this on AP questions before, is that a ribose sugar, ribonucleic acid for RNA, is going to be OO. In my head, I sing OO. That's how I remember it. But a deoxyribose, as in a deoxyribonucleic acid, or DNA, is going to have a 5 carbon sugar that is I'm sorry, I wish there was a catchier song. It's either going to be OO, OHOH for ribose, or O for deoxyribose, which is going to be the DNA. So depending on which nucleic acid you're looking at, they're going to have a different nucleotide structure with that. Also, they're going to use different bases. So what 3 bases, 3 of them, do DNA and RNA have in common? You can just say the letters. You don't have to spell them out all the way. What 3 bases do DNA and RNA have in common? Very good. Alessandra and Zara, AGC. Very good, Angel. GCA. Yes, so they're going to have that adenine, guanine, and cytosine in common. What's the 4th one that RNA has? U. Very good, Zara. RNA is going to use uracil, whereas DNA is going to use thymine. So when we're looking at these, we can categorize them in a couple different ways. We can say they're purines and pyrimidines, or we can categorize them by who they pair with. So for example, who does A pair with in DNA? T. Very good, Sandra, Zara, Angel, very good. So A pairs with T. In my class, we sing apples in the tree. A goes with T. It's what helps them remember. They know it from memory. I can just say apples, and they go in the tree. Stick that in your head if it helps you remember. But on the other hand, C with DNA is going to pair with G. So we say apples in the tree, and the kids go cars in the garage. Don't know where we heard this from. I'm sure it was like an Amoeba Sisters video or something like that. But all I know is I can say apples, and they go in the tree. And then I say cars, and they go in the garage. A, T, C, G. It works. As long as you don't have your apples in your garage. Otherwise, you're going to have a hard time. So, but the other way that we can categorize these is purines and pyrimidines. So purines are going to be 2 ring structures. So they've got 2 rings. You can see here. Whereas pyrimidines are going to have a single ring. So one purine must always pair with a pyrimidine. So A with T or U, depending on if it's DNA or RNA. And then guanine, G, is going to pair with C every time. And the reason they pair together is adenine actually will need 2 bonds, 2 hydrogen bonds to bind to thymine. So A and T have 2 hydrogen bonds. So let me make sure y'all can see that. A and T have 2 hydrogen bonds. But whenever you pair cytosine and guanine together, they're going to have 3 hydrogen bonds. So that's why they have to go together. It's maintaining those numbers of hydrogen bonds. Now, why can't they just all use T? Why does RNA need to have uracil in the first place? Like, what makes it so special? Just so you know, if you look at, I think y'all can see it. Okay, so I want you guys to go ahead and look at this picture under pyrimidines. You can look on either one of these, but on uracil and thymine, do you guys see any difference between uracil and thymine? What's the difference between uracil and thymine? It's really little, but if you go like this at your computer, you can kind of see it. What's the difference between uracil and thymine? Very good, Jess. Ooh, Jess, that's a good name. That's my name. Yes, it's an H3C. It's a methyl group, carbon with 3 hydrogens. So if you look here, you can see thymine has a CH3 group. Uracil doesn't. What that means is thymine is bigger. It's like, whoop. Uracil is only a little bit smaller, but it is smaller. And the thing about uracil being smaller is that it makes the RNA molecule smaller. So we're going to find out later that DNA turns into RNA, and that RNA is made in the nucleus, but then it has to move out of the nucleus. What are those little holes in the membrane of the nucleus so stuff can get in and out? Nuclear pores. Very good, Angel. Very good, Sandra. So those pores are like, so you know you got pores on your face? I got some. Hopefully yours are small. Mine are not. So pores are just little holes, teeny tiny holes. Thymine is too large to fit through nuclear pores. So if RNA used thymine, it would never be able to exit the nucleus. It would never be able to make protein. It wouldn't work. So instead, they use uracil, which is smaller. Cool facts, huh? You don't need to know that for AP. I just think that's interesting. Okay, let's talk about one more thing about DNA. I tell kids this all the time. I may have mentioned this in my previous DNA video, but I really want to make sure people know this because they love to throw this particular case study on the AP exam. It's called the Hershey-Chase Experiment, and it's a CHOMPS question. CHOMPS, carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur, the main elements in living things. So the deal is, back in the day, like before we had, the science we have now, people weren't sure if you got traits from your mom and your dad, if it was because you got their DNA or if it was because you got their proteins. They honestly didn't know, and they didn't have a way to know. They didn't have like the level of scientific sophistication that we have now. But Hershey and Chase did not let them stop them. What they did was they took radioactive sulfur, because if you remember, proteins can have sulfur, and they took radioactive phosphorus because all DNA is going to have phosphorus. They labeled the molecules with these radioactive isomers. So these are like little highlighters saying, whoop, here I am. And when those bacteria reproduced, the radioactive sulfur wasn't in the offspring. The offspring were completely normal. But the offspring that were marked with the radioactive phosphorus, they were glowing like Christmas trees, which meant that the DNA that had been highlighted, the phosphorus in the DNA that had been highlighted, had been passed on to another generation. And that's how they figured out that DNA is the hereditary molecule. Now, you don't need to memorize this. I tell my kids this when I tell them this story like 4 times a year. You don't need to memorize any of this. But I like my kids to be familiar with it because the AP exam loves to throw case study questions about this. You know, the ones where you've got like a full straight page of writing before it even gets to the question on the back. And so I like to go ahead and tell them this because then I think they can skim the question a lot faster as opposed to if they've never seen the question before. So that's my thinking with that. Any questions so far? I've been talking a lot and we're already 15 minutes in. Three, two, one. Here we go. Okay. All right, so let's get into the actual DNA replication. So DNA replication, can anyone tell me what phase of the cell cycle that's going to occur under? When does DNA replication? S, very good. Very good, Angel. Very good, Sandra. So, very good, Zara. So, DNA synthesis is going to occur in, that's the S phase, S for synthesis. That's going to be the DNA replication phase. And you'll notice it happens right before the final checkpoint of G2. And right before the cell splits its nucleus in mitosis, DNA replication is you have to replicate your DNA. Because if you didn't, then each cell that replicated would only have half of the DNA. Then the next generation would only have a quarter. Then the next generation would only have an eighth. Then the next generation would only have a 16th. By the time you get like 5 generations, each one only has like a 32nd of a DNA molecule. This is not Harry Potter. It's not like Voldemort with the Horcruxes. You cannot survive with everything split in 7 places. So DNA has to replicate itself. I've got this cute little GIF, and you can see there's some stuff growing, but I just think it's cool. I like to put GIFs in my slides because I think it makes them look sophisticated, but I also recognize that looking at it, it's kind of hard to tell what's going on without some labels. So let's go forward. Okay, so DNA replication has to happen before the cell can divide. So like right here, you can see this cell is going through cytokinesis and telophase. It is splitting into those 2 identical daughter cells. If you want to do that, you've got to replicate the DNA first. So for a very short time, the DNA is actually doubled, just sitting there, and that's fine. It's not transcribing anything then, which we'll get into in a minute. It's just waiting for division, waiting to go through mitosis so it can make some daughter cells, whether it needs to like heal a wound or grow or just maintain homeostasis, whatever it is. So in DNA replication, we're going to have a lot going on. So this GIF is just starting over. So the DNA is going to split open. So you can see here it's splitting open. And as it does, we have new nucleotides added to it. And so by the end, we go from having just 2 strands of DNA to having 4 strands of DNA. Here it is. Da, 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 da. 4 strands of DNA. And that's replication. We went from having 1 piece of DNA to having 2 pieces of DNA. Woo. But I do acknowledge that this is a very simplified model. And AP expects you to know certain enzymes. Those enzymes are helicase, DNA polymerase, topoisomerase, and ligase. Normally, AP doesn't really expect you to memorize certain molecules. It's only in the past couple of units that we've had to do that, like with cell communication, things like that. Even photosynthesis and cellular respiration, they didn't expect that too much. But they do expect you to memorize these molecules for this unit. You need to know helicase, DNA polymerase, topoisomerase, ligase. So let's go ahead and talk about them a little bit. I can't remember. Yeah, we'll come back to that. Okay. So helicase is going to be the enzyme that's actually splitting the original DNA apart. It unzips the genes, so to speak. So those genes get separated and they're opened up. Because I don't know if you've ever looked at like a model of DNA. You always see it in like this perfect, pretty spiral staircase cascade. But what it really is, girls, you can do this if you have long hair. Boys, you can do this if you have long hair too. So I'm going to show you something. So if you have longer hair, you may do this in class when you're bored. I do this sometimes when I'm bored. You take your hair and you just start twisting it. So DNA is twisted too. Remember, chromosomes are just chromatin that has been super twisted up. And the more you twist it, we say it has been super twisted, just like your DNA is super twisted. And as you twist it, you notice it kind of starts to kink and curl up. It has nowhere else to go. I don't think this is healthy for your hair. Please don't do this, by the way. Maybe it is. I don't know. I don't have anything. Yeah. So then you have this little nugget of chromosomes or hair, and it's super twisted up. And when it twists like that, I can't access it to do anything with it, which is really annoying. DNA is the same way. So what we need is helicase. And that helicase is going to be responsible for taking that. I shouldn't have dropped it yet. It's going to take that super twisted up DNA and it's going to split it apart. And that way, both of the strands can be accessed. But then it's super twisted and coiled. So we have another enzyme. I'm going to make my face smaller now. Topoisomerase. So if helicase is doing the actual separating of the strands, topoisomerase is the enzyme that is relaxing this supercoiling. So like when the DNA gets like my hair and you just twist it and twist it and twist it until it coils up. Topoisomerase is the enzyme that's actually going to loosen that super coiling, that super twisting. Good so far? Got 2 more enzymes. Does anyone have any questions? I just played with my hair in front of y'all for like 2 minutes straight. So surely someone has a question. 2, 1. Okay, I'm going to keep talking then. So once you get everything all untwisted, so we've untwisted it and we've separated it, where is topoisomerase used in the process? So it's going to follow directly behind helicase. So like helicase comes through, and then topoisomerase is going to relax it all. They're pretty much happening side by side. Does that answer your question? Great deal. Okay, so then we're going to have this enzyme called DNA polymerase. DNA polymerase, so you can see for a second when they unfurl, I'm going to wait. Okay, so in the GIF, you can see when they unfurl at first, they're empty. So that means they don't have hydrogen bonds holding them together. Do molecules like it when their bonds are missing? Are they happy about that? No, they don't like it. They have a certain balance, and that goes back to basic chemistry, which is not my job, so I'm not getting into. But bottom line, they don't like it when their bonds are missing. So as a result, they will reach for anything they can grab. And polymerase is like, I got you. So polymerase swoops in. It's polymerase. It makes polymers. Polymerase. Polymerase. It makes polymers by adding those nucleotides to those open strands. So you can see like down here, these strands have already been filled in. Okay, the helicase and the topoisomerase have come through and they've moved to the right over here. But the polymerase is the enzyme that has come in and added this new layer of nucleotides. And as a result, it now is like a healthy strand of DNA forming. This one is also becoming a healthy strand of DNA, but here's the issue. We haven't really talked about this. DNA is what we call, the process is semi-conservative. What that means is we keep half of it and then we use it as a template to make the new one. So like you can see here, the original strand is red. It's kind of light, but the new one is pink. And so we have half of each strand is from an old strand. But then half of each strand is a new strand. So it's semi or half conserved, saved. I put this on here because people used to think it was conservative or dispersive, which were patterns that look like this. They have since been debunked. It is not conservative. It is not dispersive. It is semi-conservative. But the other deal about DNA replication is that it happens on anti-parallel strands. So what I mean by that, so if someday when you're in organic chemistry, or even I think freshman chemistry, they probably teach you this, you have to learn how to label your carbon atoms in your molecules. And the reason for that is it's how you name molecules. So it's like whenever you see weird names in chemistry, like 1,2-polydiol or something weird. I don't know. I haven't been in chemistry in like 8 years, guys. Work with me. But it'll be like 1,2 something something. And the reason they do that is they're listing the specific carbon that has a feature. So like it's really light, but you can see on here, these carbons, we have a 2' carbon, which means this one up here must be 1. This is 2'. This is 3'. This is 4'. This is 5'. That's all that 5', 3' nonsense means. It's talking about the different carbons on the pentose or 5-carbon sugar. And so we say the DNA replication runs 5' to 3'. What we mean by that is when it's being built, it's going to go in this direction. So you can see my little red dot. It's going to start at a 5' and work down towards a 3'. So if this strand is 3', these are anti-parallel. That means they run in opposite directions. Think of it like driving down a highway. When you drive a car, you can't drive on whatever side of the road you want. You get tickets for that. So instead, you have to drive on the right side of the road, and the person driving the opposite direction also drives on their right side of the road. You're both going different directions. It's parallel because they're next to each other. It's anti because they're going the opposite way. So for example, if this strand up here has 3' on one end, so that means that it's 3', it's got the bottom of this pentose sugar facing out. That means that down here must be the 5' end. So DNA replication is going 5' to 3'. That means it's working down. So it's matching with that 3'. That's a 5' because it's anti-parallel. So this is what we call the leading strand. That leading strand is going continuously replicating. The DNA polymerase is just like boom, 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 boom. It's adding nucleotides, it's doing what it's supposed to do. But then there's a problem, because there always are. So then we have this strand over here. So if this one is 3 to 5, that means this one must be 5 to 3. DNA replication likes to happen 5 to 3. So that means it's got to start down here on the 3' end, and it's working backwards. As a result of going backwards, it can't just do it all at once. It's kind of like if you are walking backwards. You can't just walk backwards. You have to stop, look over your shoulder, see what you're missing. Stop, look over, see what you're missing. RNA or DNA polymerase is doing the exact same thing here, and so it stops. And as a result, it forms these things called Okazaki fragments. Everyone always remembers that word. So they may not remember mitochondria as the powerhouse of the cell, but they will remember Okazaki fragments. It's the word. People just remember it. But so these fragments are in pieces, and that DNA then isn't sealed together. It doesn't like that. So instead, we have this last enzyme. It's called ligase. Ligase is like a glue that actually glues those discontinuous pieces together. And then at the end, you've got your pretty semi-conservative DNA replication. I just talked at you a lot about a very complex topic. What questions do you have? Five, 4, 3, 2. Can you repeat what topoisomerase is? Yes, of course, Oscar. Topoisomerase, all it does is relax the coiling of DNA. So you know DNA is coiled up super tightly as chromosomes, so kind of like my hair is here. I'm twisting it super tight, so tight that it actually starts coiling on itself. All topoisomerase does, it's like a straightener. It comes through and releases that supercoiling so that it's then a straight line again. That's all it is. Any other questions, guys? All right, I'm going to move on, but if we have any questions, hold them till the end and I'll definitely answer them. Maybe like save it in the chat box or go down on the bottom where it says ask a question. And that way it's still on my mind and it doesn't get lost and we can pull it up at the end. All right, moving into the big game now, the central dogma, aka gene expression, aka protein synthesis. You will see here that we start with DNA, we create RNA, from there we create a protein. So let's see how this works. Okay, this whole idea of gene expression or the central dogma started with this whole one gene, one protein hypothesis. So we all have genes. We get them from our parents. I have a gene from my parents that, not from my parents, from my dad that made my eyes this lovely shade of blue. It also made me kind of blonde, but not really, you can tell. It gave me certain features. You probably have the same thing. If you have red hair, you can thank your parents. If you have no hair, you can thank your parents. If you have double jointedness, I don't know, you can thank your parents. All of those things are controlled by a gene. But it's not just those features, it's also innate things we can do, like the ability to make the saliva in your mouth with the enzymes that break down your food. That's thanks to a gene. The ability to create create the membrane in your ear, the eardrum that lets you hear. You have a gene for that. You have a gene for literally everything. It's a lot. And those genes just keep going and going and going and going. But the idea here in the one gene, one protein hypothesis is that every gene in your genome, that is your complete set of DNA, is going to create one protein. I will tell you, this has since been debunked, but for the purpose of this, it's a good model. So for example, let's say I'm looking at a section of DNA. So I have taken my DNA, sorry, I'm looking for a pen. I forgot y'all were watching. I have taken my DNA, I have synthesized it, and I want to make the protein that makes my skin freckly. I want to make that protein. So I've got my DNA. And what I've got to do is I've got to find the place in the DNA that controls my freckles. And I am going to transcribe that freckle gene. I don't want to transcribe these genes. They have nothing to do with freckles. I just want to get the freckle gene. So I'm going to do this through gene expression. Again, there's 2 steps to it: transcription, translation. Transcription is where we turn DNA into RNA. Translation is where we turn RNA into protein. So for example, let's say instead of a freckle gene, I can look at a chromosome and it'll have this little band, this place that's called a loci. It's Greek for location. Go figure. But this loci, or loci, depending on how your teacher pronounces it, doesn't matter as long as you spell it L-O-C-I. Every gene you have can be mapped to a certain location. Everyone has that. And they're all in the same location unless you have a weird mutation. But so for example, on chromosome 17, everyone has a gene that creates a protein that will transport serotonin. Serotonin's a thing in your brain, makes you happy. You want to have that. That gene's not doing what it's supposed to, it can have some serious side effects on your mental health. So let's say I want to take this serotonin gene and I want to make sure it goes through gene expression so that I can have a clear and happy mental state of mind. So let's take a look at how that's going to work. We're going to start with transcription. Get the transcript. Ta-da-da. I'm trying to like mirror him here. So what you've got when you're doing transcription. So in transcription, the DNA is going to be unwound. That's going to be the helicase and the topoisomerase. Again, just unwinding everything. But now when it's unwound like that, instead of saying leading and lagging, we are going to say antisense and sense strand. I am going to look at the antisense strand. The sense strand, we're not doing anything with it. It's just going to kind of hang out. But that antisense strand, you can see here, what's happening is this DNA strand on here is getting turned into RNA. So every C is matching with a G, every T is matching with an A, every G is matching with a C, every A is matching with a, what do the A's match with? A in RNA? If we're making RNA, what is U? If it was DNA, but we're not replicating DNA here. No, it's good, Sandra. We're not replicating DNA here. We're making RNA. And we're making a transcript of RNA. A transcript is a copy. So here's the way that I think of it, gene expression. DNA is like the president. It lives in the White House, and when it leaves, it has a lot of security because if something happens to it, we are in big trouble. DNA doesn't leave our nuclei. It doesn't. It's not supposed to, because if we lose it, that cell will die. So instead, what DNA does is it makes a photocopy. So like, pretend I've got, so like, y'all can see this behind me. Let's pretend that I highly cherish this picture of a cactus that I painted myself that is not the highest quality. Let's pretend it's like the Mona Lisa level of art. If that was the case, I would never let it out of my sight. I would never take it out of my house. I would want it completely protected. But I would still want to share it with people because I'd be like, look at my awesome painting. So what I would do every time I want to show people, I'd take a picture and show them the picture. RNA is like that. It's the copy of the DNA. It's something that is safe to leave the nucleus because it's just a photocopy. It's not going to hurt if it gets damaged. We've got the original still. So this DNA gets turned into RNA. So those DNA strands, so you can see down here at the bottom where it says DNA strand whoa, what are we doing? Oh, sorry, my computer was being a little weird. So like here it says ATG, CTT, CGA. And that's going to match with another DNA strand that says TAC, GAA, GCT. But if I take this top strand and turn it into RNA, or if I take this bottom strand and turn it into RNA, sorry, it's going to be the same, except every T is going to be replaced with a U. That's all it is. We're just making RNA. And we're going to do this. We're not using DNA polymerase this time because DNA polymerase is for DNA nucleotides. Instead, we're using RNA polymerase because it's for RNA nucleotides. Are DNA nucleotides and RNA nucleotides the same thing? Are DNA nucleotides and RNA nucleotides the same thing? Yes or no? No. Very good, Sandra. If you remember, DNA nucleotides have a deoxyribose. RNA nucleotides have a ribose. Remember, ribose goes O-O. Deoxyribose goes O. Also, RNA doesn't have thymine. It just has uracil. DNA doesn't have uracil. It just has thymine. Remember that, guys. DNA and RNA nucleotides are completely different. So we're going to use RNA polymerase because we're making RNA. If we were making DNA like we would with replication, then we would use the DNA polymerase. But we're using RNA polymerase. So once you've got your RNA, that RNA can't just like go out into the world. So again, it's like the president, except this is a copy of the president. This is like the president's ambassador. But if you were the ambassador to the president, you would probably still need some security, right? Do you think you would need security if you were the ambassador to the president? Yes, definitely, Vanessa. Very good. So if you were the ambassador to the president, you would need security. You would need some refinement before you went to like negotiate with all the other countries. RNA is the same way. Before it can go and do its job out in the cell, it's got to have some refinement. And this is what that refinement looks like. So RNA is made of two sections. We have exons and introns. Exons and introns, they're the exact same thing they were before. They're just RNA nucleotides, A, U, C, G, in replicating patterns. But whenever RNA is processed or refined, those introns are actually removed or cut out. There's a lot of theories behind why this happened and how they can recombine to create other genes, but I'm not going to get into that right now. I may at the end if we have time. The main thing you need to know, introns are removed, exons are preserved. Introns are removed, exons are preserved. Keep that in your head. Once you get rid of all these introns, then it adds a 5' cap. So this is going to be a little cap. It's also called a methyl cap or a GTP cap. Doesn't matter what you call it, as long as you call it one of those three. You can even probably call it a cap, and it's fine. But this cap sits on the 5' end of the RNA. But then at the end, we have this poly A tail. Here's how you should think of it. So this RNA, once it's been spliced, this is the process of removing the introns and keeping the exons and adding the cap and the tail. Once it's been spliced, this RNA is going to leave the nucleus. It was created in the nucleus, but it's now going to go out into the cytoplasm so it can make the protein. The cap is kind of like the driver of a limousine. It's taking the RNA where it needs to go, which will be to a ribosome. The poly A tail, that's like the security. They're there to make sure that the RNA doesn't get messed up. Because once that RNA enters the cytoplasm, it has a chance of getting chomped up by enzymes called exonucleases. And those exonucleases are just doing their job. Their job is to chop up RNA so that it can be processed and recycled to make new RNA. So this poly A tail It's like it's chomping on the back, but it doesn't actually get to the RNA that's needed for the next step. So then the RNA leaves and goes into the cytoplasm so it can do translation. Before I get into translation, does anyone have any questions? Any questions? All right, take your silence as a no. So, translation. This is the second step. This is the part where we turn the RNA into the protein. So what's going to happen is you're going to have this mRNA, this strand. So I'm going to kind of go here so I can see. So you've got this mRNA. It's made of those nucleotides. You can see it's got the cap. It's got this long yellow tail. That's all the poly A. That's that A tail. So it can get chomped up by exonuclease, but it isn't going to hurt it. And what's going to happen is the ribosome is going to kind of chomp on the RNA, and it's going to pass through it like a conveyor belt. And as it does, it's got this new type of RNA. It's called tRNA or transfer RNA. tRNA is going to read each section of RNA and connect it to a specific amino acid. Those specific amino acids are going to correspond based on a triplet code. You're going to use a special chart for that. We're going to take a look at that in a moment. That special chart on here is going to make different amino acids. When you put a bunch of amino acids together, what do you get? What do amino acids make? Tell me. Protein. Very good, Zara. Very good, Vanessa. A bunch of amino acids strung together, that's the primary structure of protein. That's it. I had a gene where I wanted to make something. My mom gave me a gene for, actually my mom didn't give me a lot of phenotypical genes. My dad gave me a gene for blue eyes. He gave me that gene. And that gene is expressed when my DNA is transcribed into RNA. And then that RNA is translated into a protein. And that protein builds my blue eye. Boop, boop. That's what it is. That's this whole light bulb. Every organism goes through transcription and translation, and that's how it builds itself. That's how the DNA is read and used in order to actually produce something. Do we have any questions about translation up until here? Three, two, one. All right, I'm going to keep going, but if you have questions, let me know. So a little bit further on this translation process, to go more in depth into it, it's got 3 phases. They're called initiation, the start, and this is going to be when your mRNA is actually going to bind to that ribosome. So you've probably heard before, ribosomes have a large subunit and a small subunit, and the RNA is literally just going to pass through these 2 subunits. That's kind of going to hold it in place so that it can have whatever it needs done. Then we're going to have elongation. That's the second step. In elongation, that tRNA, that other type of RNA, which is also made in the nucleus, is going to drag different amino acids that match with the mRNA. And as it does, it's going to grow this long, long chain. That is a chain of amino acids. That's your protein. Until it reaches something called a stop codon, it's just going to keep going and producing this longer and longer and longer protein. You can see in the GIF over here that it's doing it. It's moving down the mRNA. The tRNA is bringing those beads, which are making the protein longer and longer and longer until it gets to the end. And then that protein can be released. And then the RNA is just recycled. It gets chomped. And it goes and it can make new RNA. But now you've got the protein that you need. Ta-da! Are the 3 letters the codons? Yes. So whenever we have an mRNA strand, it's always the number of nucleotides is always going to be in multiples of 3. Because each 3 codons, each 3 nucleotides is 1 codon. That codon is then going to correspond with a specific amino acid. That being said, multiple codons can code for the same amino acid, because there's only 20-something that living organisms on Earth at least use. Very good question, Sam Sarabia. Okay. Any other questions about translation? Okay, I'm going to go ahead and click on, but let me know if you think of something. So this is the summary of how it's working. You've got your DNA, and that DNA is going to be transcribed, and that's going to create RNA. To create that RNA, you have to process it by removing those introns. They get spliced out. Then you have a complete RNA. That RNA then is translated into a protein. It's as simple as that. If you understand that concept, you will do fine on the AP test. You just need to understand that the point of this is to express your genes. It's that if you have a gene for, I don't know, a widow's peak, that gene is transcribed as RNA. That RNA is translated to protein, and you have a protein that causes that shape on your forehead to form. If you have a disease, that's most likely because you have a gene that is producing some protein that is causing harm, or that disease could be because you are missing the gene and now it's not producing the protein. Any questions? Three, two, one. Okay. Oh, actually, I think that's it, y'all. That's my last slide. Wait. Yep. That's my last slide, y'all. What questions do we have? How are we feeling? What do we think? Talked about DNA structure. We talked about DNA replication. Talked about the cell cycle a little bit again. We talked about the central dogma / protein synthesis / gene expression. We talked about transcription and translation. What questions do we have? Five, 4, 3, 2, 1. What is central dogma again? Okay, so Vanessa, all the central dogma is the process of transcription and translation. Central dogma is just another word for protein synthesis, the process of making protein, or gene expression, the process of expressing your genes as proteins. Central dogma is just another fancy phrase biologists like to use to describe those two processes. Does that help, Vanessa? Awesome. Okay, Ariana, repeat the initiation step. Okay, initiation, it's relatively straightforward, but it's also a little complicated. I didn't put a diagram on here. I probably should have. But remember, RNA is going to be made in the nucleus. But when that RNA leaves the nucleus and goes out into the cytoplasm, it's going to drive around until it meets a ribosome. When it meets that ribosome, let me see. Okay, so when it meets that ribosome, what's going to happen is that ribosome is going to be in 2 separate pieces. But those 2 separate pieces are going to clamp down on the mRNA. So you can see here how the mRNA is kind of running through the center of the ribosome. That's initiation. That's all it is. It's when the RNA connects to the ribosome. Does that help, Ariana? Okay, great deal. And then I just see Madeline, you asked a question a second ago. Are the tRNA in the ribosomes? tRNA are not in the ribosomes. They float around in the cytoplasm. Or this can also happen on endoplasmic reticulum because they have ribosomes on them. But they're not in the ribosomes. They float around. They attach for a second to drop off the amino acid, and then they float away again. Does that answer your question, Madeline? Very good. Okay, Oscar, when is tRNA made? So tRNA is probably, I'm not 100% sure, but I would imagine tRNA, it's an RNA. It's probably made in the nucleus by the DNA. It's another gene. You have a gene that makes tRNA, and a different gene for each amino acid that the tRNA carries. And so it's just going to be made through transcription. It's just never going to be translated because it never becomes a protein. It stays an RNA. Does that make sense? Awesome. All right, what other questions do we have? We got another couple seconds if we need them. Five, 4, 3, 2. Is there a difference between RNA and mRNA? Very good question, Sam Sarabia. So mRNA is RNA. When it has that M in front, that M, I don't think I forgot to say this, that M, so this is a really good question. That M stands for messenger. So messenger RNA is just a type of RNA. tRNA is another type of RNA. That T stands for transfer. They're just different types of RNA. Does that help? Awesome. Last call. What have we got? Ask me questions. Ask me things. Three, 2, 1. Best steps to memorize all this? Flashcards. Get a Quizlet deck. That's all I can tell you. You will never be asked to draw it, although I will say if you can draw this from memory, that does help. But in the end, it's just flashcards. That's the only way I have ever been able to do it. Sometimes I can do it if I rewrite my notes by hand all the time, but that just depends on how much time you have. Does that help? I wish I had something more concrete, like do this. But that's all I've ever been able to do to make it work. What other questions? Five, 4, 3, 2, 1. Last call, y'all. 3, 2, 1. 0.5. All right. I guess we are done, y'all. Thank you so much for coming. I hope this was really helpful for y'all. Like I said, best way to memorize this is if you can draw it from memory, great. Flashcards, though. Everything is about flashcards in the world. So get yourself lots and lots and lots of them. Thank you so much for coming, you guys. Like I said earlier in the big plug, all this information is free now. It's completely accessible to you guys on the website. So get it. Sandra, thanks for coming. Woo. Sarah, glad you were here. Madeline, you too. Thank you, Consuelo. Whoop, whoop. All right, y'all. I'm going to go to bed now because it's my bedtime because I'm old. Thank you for coming, guys. See y'all later.